All right, welcome back, zoologists. We are now finally going to get into the arthropods, one of my favorite groups. They are hugely diverse, and hopefully I can convince you of that. This is one of the biggest groups. It'll be the biggest clade that we cover of the entire semester. But we won't cover it in that much detail. We won't go in as much detail into this as we do other of the phylogenies, or other clades, sorry. So this is the phylogeny I've been showing you all semester. So clearly we're still within the Bilateria because that's they include everything that we're going to cover this semester, with a few exceptions. But we've only recently gotten into the Ecdysozoa, so hopefully you'll recall that these are all organisms that molt in order to grow, and that's because they have a hardened external exoskeleton, and that molting is mediated by the hormone ecdysone. We already have discussed the nematodes and the onychophorans, and now we're going to get into this group here, the Arthropoda. So let's talk about some of the big pictures, like major evolutionary innovations in the group, and then we'll work our way down. I should probably introduce the group a bit first. So this group includes things like spiders that are very familiar. These are barnacles. Isopods, so you're actually seeing a crustacean that's right here on the side of this fish. So we're not talking about the fish itself, but this thing that stuck to its face. And of course all kinds of insects, two of them here, predator and prey. Crabs and all kinds of things that we like to eat, including lobsters as well, lobsters and shrimp and crawfish and that kind of thing. And as mentioned, this is one of the biggest groups, and one of the reasons that the arthropods are one of the biggest groups is because of the hexapods, so basically the insects plus the spiders. So there are thousands and thousands of species within this group, and we'll just touch on some of the highlights for this group. But if you really want to have some fun, you should definitely take entomology, the study of insects. Okay, so the name Arthropoda comes from jointed foot. So this group is characterized by having lots of appendages, many of which are specialized for specific purposes, and they're jointed, so they have more than two parts to them. That makes them incredibly important and useful because now not only do you have lots of appendages that can move in all kinds of directions, which gives you greater mobility over where you're going, greater directionality, greater mobility. It gives you much greater ability to actually capture prey, to look for mates and to hold on to mates, to hold on to your young, to defend yourself against predators. The utility is almost boundless. And this picture right here sort of highlights that very diversity. So you see lots of different kinds of appendages, everything from antennae, which are of course used for sensing the environment, to chelicerae, so these are modified into claws that are used for both defense as well as prey capture. You have all kinds of walking appendages, and underneath you would see swimmerets, and then you have a tail that's also used for swimming, so all kinds of appendages. So it's important to note that these are coelomates, true coelomates. They're also protostomes, and they have well-developed organ systems. Now we're going to go into this in more detail in just a few minutes, but you will see on the inside that all of their organs are well-developed, and from now on what you're going to see is an extension of that development, so really high-order specialization for the most part. In pretty much all the organ systems. So as we work our way all the way up the phylogeny, you're going to see really incredible development of the brain and subdivisions of the brain as they govern different parts of the body. You'll see all kinds of specializations in the anatomy as well, and digestive. So they are, of course, characterized by having this exoskeleton, and so it's a cuticular exoskeleton, so it grows out from the cuticle, the outer portion, and it's composed of chitin and as well as protein combined, so it's a very strong substance. This exoskeleton is a very huge innovation, big deal, because it allows for, especially in combination with the jointed appendages in this case, because it allows for the muscle attachments. And so now you have, instead of having to depend on a hydrostatic skeleton, you have this hard protective outer coating, so it's protecting everything in your body, right? It's like you're wearing a suit of armor, right? But at the same time, you also have muscle attachments, and so that gives you really great ability to move your body parts around and to have really extensive appendages sticking out that you can then mobilize. Can't really do that with a hydrostatic skeleton as well. So previously we had talked about metamerism and used as the example for that the annelids. So these guys exhibit metamerism as well. So remember, this is serially homologous segments. But in this case, some of those segments are fused into body regions that are called tagmata. And so we haven't really seen body regionalization as clearly in the previous organisms as we're going to see now. So you'll have a really specific, let's say, head or cephalothorax, and then abdomen or subdivisions of the body. And those subdivisions we will call tagmata. And again, in some cases, it's because of repeated segments that are fused together, like you see in the upper body, if you will, of this crayfish. I've already said this 100 times already. Lots of appendages, lots of them are specialized, and we'll talk about those specializations as we go through each group. Oh, but I'll highlight some of them here. So you'll see not only lots of legs, but legs that are actually specialized for different purposes. So sometimes some are smaller, some are bigger. Sometimes they have special pockets on them, say for carrying pollen around in the case of these bees, which is what's happening here. You'll see all kinds of appendages for defense as well as prey capture, and also for breathing. So these are limbs that are attached to gills that are on the inside. You'll see all kinds of modifications to the first set of appendages into things like fangs. You'll also see in a lot of these guys, the mouth parts have a tendency to move sideways, both the maxilla and the mandible, as opposed to up and down. And I find that simultaneously really fascinating and disgusting all at the same time. So it's hard for me to wrap my head around mouth parts moving in this direction. But we'll see some examples of this, and we'll see some videos in class. Okay, these guys are also going to have a real well-developed sensory set of sensory organs. Insects are highly specialized in terms of their antennal sensory, so chemosensation as well as vibratory or vibrations, tactile sensations. But they also have really well-developed eyes and all kinds of adaptations like wings and other kinds of structures that are highly innervated. And so we'll see some examples of that. So not only will you see a huge development of the nervous system throughout the body, powering. So remember, your brain is the central nervous system. It's the central core or computer that governs the whole body, and it's running the show. And so you're going to see greater innervation throughout the body, also powering these specialized appendages and structures. But you're also going to see greater development of different aspects of the brain because of how those parts of the brain are governing parts of the body. And that's something that you're going to see as we go throughout the rest of the semester, the development of the brain and different parts of the brain. Now, one thing that's not, I wouldn't call this a major innovation here, it's more of a minor one in the grand scheme of the phylogeny, but it's important to the arthropods, and that's this tracheal system of respiration. So it's really an interesting, independently evolved respiratory system that's not unlike our lungs, but operates differently. And we'll talk about this in a little bit of detail. So this is just an example of a grasshopper, but most arthropods have this system, and the Onychophora do as well. On the sides of their body here, where I'm pointing with the cursor, at each intersection of each segment, there's a little hole, and there's holes on either sides of the body. And these are spiracles. And so they can open and close them muscularly. They have the ability to do that. And these holes lead into these tubes that go into the body. So here's the spiracle, the opening to the outside of the body. So you can see it goes into these little tubes inside the body. And they don't show this very well, but these things are highly vascularized. And then they ultimately interdigitate with the muscles. So what's happening is air flows into these tubes, And they actually can make muscular constrictions to sort of pull up a current to allow the air in and out. And because it's highly vascularized, um, oxygen can then go through the very thin membranes of the tracheal system and into the circulatory system to power the muscles. So this is showing you just a simple diagrammatic view of a kind of a grasshopper-like organism. And you can see these little portholes, these spiracles on the sides of the body, both sides, and how they basically go into this tubular system, this tubular network throughout the body of a grasshopper. So this is one of the ways that they can respire and get oxygen, or engage in gas exchange, I guess I should say, gas exchange throughout the body. Many of them, but not all arthropods, have a compound eye. This is a very complex eye structure that gives them quite acute eyesight. They can see forms very clearly, and they have really good depth perception. That's especially good for organisms that are flying. So we're not going to go into the gory details of this eye, but suffice it to know that a compound eye is called that because it's made up of hundreds of these cells, these ommatidia cells. And these cells are composed of a lens that allows them to focus a bit. They have pigment cells and retinal cells inside them. And then many of these are combined into this eye. And sometimes you have multiple compound eyes, not always, but sometimes, on certain arthropods. So they have a very good eyesight and very good depth perception. Okay, so aside from some of those morphological features that are unique and special to the arthropoda, they have some other aspects that are also very important to their success on the planet and to their evolution. So two of those features are complex behaviors and trophic breadth through metamorphosis. So we could go on for years talking about both of these topics, but when we say complex behaviors, we mean that arthropods can exhibit, and we'll talk about examples of this in class, but all kinds of really interesting behaviors that facilitate their evolutionary and ecological success. So for instance, bees, this is a famous example. So bees are able to do what's called a waggle dance back at the hive, where scouts can come back and through a dance that they give to the other members of the hive, they can describe where food resources are. So by doing this particular dance in a particular way, and I'll show you videos of this in class, they can communicate very accurately what direction, how far away flowers are that they can then feed on. This is an example of horseshoe crabs that are in a mating frenzy that happens once a year along the east coast of the United States. So these are one of several organisms that actually have these mating congregations that are cued by the environment, and they come together and mate and then lay eggs in the sand, actually thousands and thousands of eggs on the land. And we'll talk about that mating behavior in class. They also have all kinds of interesting mimicking behaviors, particularly in the insects. This is a diagram that's meant to be showing you with the flashing patterns of fireflies. And what it's supposed to be showing you, these letters indicate 6 different flashing patterns that fireflies do. And what's especially interesting is not only that fireflies have these species-specific flashing patterns, which they do to try to attract mates, but there are other insects that will mimic those flashing patterns in an effort to try to eat or lure individuals in and eat them. Sneaky little devils, aren't they? And they exhibit all kinds of really cool other mimicking behavior as well, which we'll talk about in class. So these are caterpillars that can modify their bodies and or are just cryptically colored to look like snakes or predators that most organisms probably wouldn't bother tormenting, and they manage to get away from predation because of this mimicry. So the element of mimicry in insects is just unbelievable. So those are that's just a sense of some of the complex behaviors I'm talking about. I'll explain in class how these facilitate survival. But they also exhibit trophic breadth through metamorphosis. So you guys are probably very familiar from a really early age that all kinds of insects go through metamorphosis. The most famous example is, of course, that of butterflies. So everyone learns really early that butterflies lay eggs, they turn into caterpillars, they feed on plants for a while, then they pupate, they go through this chrysalis stage and ultimately transform from this caterpillar into a butterfly. Well, lots of arthropods go through metamorphosis where they go either through a really dramatic change from a caterpillar to a butterfly, in this case through several morphological stages. This is what shrimp do. And in some cases they go through a metamorphosis that seems potentially less dramatic. They kind of come out looking a lot like an adult, but maybe they just lack wings and they're smaller. But then they go through stages up until they look like an adult. Either way, this metamorphosis is really a huge evolutionary innovation because at each of these stages, they're utilizing different resources. And so by utilizing different resources, they're actually dividing and conquering among themselves and reducing competition among the different stages. So for instance, you have caterpillars that are eating, say, the leaves of a particular plant, but then the adults will be eating, consuming nectar, let's say, of the flowers of either that same plant or probably some other plant, but they're not consuming the leaves. And same thing with these stages of these different organisms. So they're usually eating, each of them is eating something different from the next stage up. And so that way they can capitalize on the resources around them without hopefully depleting all of them around them. And that has led to significant success on the planet. Okay, so let's talk about the groups of arthropods that we're going to cover in class. So there's 4 subphyla of groups. So we're going to talk about the Chelicerata, the Myriapoda, the Crustacea, and the Hexapoda. I'm not concerned that you know these terms up here, the Mandibulata and the Pancrustacea. Don't worry about those. It is important to note that the Crustacea and the Hexapoda are sister taxa, and those are sister to the Myriapoda, and the Chelicerata are at the base. Now, these characters that are on this tree are hugely important, and this is a great concept map way of displaying them. So if you could memorize these, that would be great. Some of these are more important than others, and I'll point those out as we go along. But something I do want to point out right here is that notice that the sister relationship here between the Crustacea and the Hexapod, that's based in part on morphology, but also on shared derived DNA sequences. So let's go into each of these groups and we'll start at the base. So the subphylum Chelicerata includes things like horseshoe crabs, arachnids, mites and ticks, organisms like that. They're characterized by some 3 main features, and these are similar features that we're going to be hitting on for each of the arthropod subphyla. So they have 2 tagmata, or 2 body regions, that are referred to as the cephalothorax and the abdomen. So you see the cephalothorax here, and then here's the abdomen, and the tail is part of that abdomen. Sorry. They have lost antennae, so they have no antennae present. And the first set of appendages are referred to as chelicerae, and they can be used for different functions, and let me show you some examples. But the first set are called chelicerae. So here's some examples of some chelicerates. So spiders, ticks, and this is a mite. So again, with this cephalothorax and abdomen, so the cephalothorax is the anterior portion, and the abdomen is the posterior portion. In the case of things like ticks and mites, it's really hard because their heads are really small, and often they're adapted for piercing into bodies. But basically you have the cephalothorax, so cephalo means head, so cephalothorax and then abdomen. Okay, so they have 6 pairs of appendages on the cephalothorax, and it's stated this way because sometimes you have specialized appendages that are elsewhere. but basically 6 pairs of cephalothorax appendages. The first set are called chelicerae, as I mentioned, and they're modified for different functions, depending on which chelicerates you're talking about. The second set are referred to as pedipalps, and they're frequently used for reproduction, but they can be accessory feeding organisms. And then there's usually 4 pairs of walking legs. And in this guy, you can see it clearly. 1, 2, 3, 4. Here's the 4. Let's look at some up close. So here's a typical arachnid. You have the 4 pairs of walking legs. The second set is right here. These are the pedipalps. And in the first set are these chelicerae. And in the case of arachnids, many times these are modified into fangs. Not in all, but in some. And here's a horseshoe crab turned upside down. So here you go. These are the chelicerae, these top 2, front 2. They're used for feeding, but they can be used for reproduction as well. Second pair of pedipalps, and then 1, 2, 3, 4. 4 sets of walking legs. Now these are some sensory appendages here that we're not going to go into. So these guys lack mandibles. They don't have mandibles. They have maxilla, but they don't have mandibles. And they have no antennae, which we mentioned on the previous slide. We'll go into additional morphological features later in class. Oh, sorry, no, we're not. We're going to go into them now. Now, within the Chelicerata, okay, so these are the features that unite all the chelicerates together. We're going to talk about 2 different classes, so the Meristomata and the Arachnida. The Meristomata are the horseshoe crabs. By the way, this term is derived from Mero is Greek meaning thigh, like leg, thigh, and stoma always means like hole or opening. And so thigh opening or thigh, and actually it refers to thigh leg. So they have appendages that can serve on one end of them for feeding and on the other end of them for swimming or for walking. So anyway, thigh leg. It's very odd. The arachnida are spiders, scorpions, ticks, and mites. So I'm not going to go into great detail about the characteristics of the horseshoe crabs because they're pretty characteristic. They almost look like a modified trilobite, and they all look just like this. And there's really nothing in the way of species diversity. So there it is. In the case of the arachnida, though, there's a couple of things I want you to know. One is that they respire through book lungs, which you can see right here, and they're called that because the different layers of the lungs look rather like leaves in a book. And by the way, going back to that horseshoe crab picture, here we go. So they have book lungs, or book gills, that they respire with out here. Okay, one structure I want you to commit to memory are these, the Malpighian tubules. So these are an extension of the excretory system, okay? They are outpocketings of the large intestine, though, and what they do is they function to take water out of the digestive system and hold on to it. So many of these are organisms that either live in very xeric environments, so dry environments, or they just have a tendency not to drink water, so they only get their water from their food items. And so what the Malpighian tubules then do is they extract water from the intestine before it leaves the body and maintain that to help balance, to maintain water balance, okay? So yes, they're outpocketings of the digestive tract, but they are an excretory organ that helps to maintain water balance. I feel like I'm hitting this and nothing's happening. Okay, with luck, here we go. Okay, so you know that the arachnids have many simple eyes, sometimes upwards of 8, and they have lots of sensory setae, so sensory hairs. So here you can see the eyes. Generally speaking, these ones are compound and these ones are simple, but there's variations on that theme. And then here on the scorpion, you can see all these sensory hairs that are all over their chelicerae and actually on the rest of the body as well. And so these give them the ability to really sense small vibrations and air currents, and to chemosense as well. Okay, let's go into the second subphylum. So the second subphylum are the Myriapoda. So myria meaning myriad, and poda meaning feet, so lots of feet. So these are the centipedes and the millipedes only. They have two tagmata, head and trunk. Pretty simple and pretty easy. They have lost the compound eye, so they have very simple eyes. They have not very good eyesight, so they're mostly sensing their environment through chemosensation. And they do have antennae, though, so they do have some tactile sensation as well. So here are some examples. Here's a centipede, here's a millipede. So two tagmata, head and trunk, pretty simple. Here's the head, the rest of it's the trunk. As you can see, and they're like classically known for this. They have paired appendages on all segments, and the legs are uniramus. So you can see that they have lots and lots and lots of legs, but the idea that centipedes have 100 feet and millipedes have 1,000,000 feet is actually completely incorrect. You can see each of their segments here, so they do exhibit a bit of metamerism. They're not fully metameric within, but they're largely metameric. This term uniramus, you'll understand a bit in a few slides from now. Or you'll understand a bit better. So uniramus means they're unbranched. It doesn't mean that they're not jointed. They're jointed appendages, they're just not branched. They don't have different sides to them. I'll show you branched appendages when we get there, but these are not branched. So like it says, though, there are paired appendages on each segment. There's one pair of antennae. You can see them here in the front. And these guys have mandibles and either one or two pairs of maxilla. So this is the thing about all these arthropods. They got not only a lot of appendages, but they got a crap ton of mouth parts as well. And I'll show you videos in class. Lots and lots of mouth parts. So we're going to cover two classes, the Chilopoda and the Diplopoda. So the Chilopoda are the centipedes. I always remember this as they both start with C. These guys have dorsal ventrally flattened bodies, so they kind of look like they've been squished a bit. Nearly every segment has one pair of legs, so one on either side of the body. The first pair of appendages are modified into venom claws. So let me show you a picture. Oh, and they have two maxillae. So here you go. If you turn them upside down, here are the venom claws. So these guys are pretty badass. You probably don't want to pick them up and handle them too much. Now, the Diplopoda are the millipedes. So it's diplo because, they have a cylindrical body. And nearly every segment has two pairs of legs. So if you look closely, that's what's happening here. Is there's two on each side, right? So 4. They only have one pair of maxilla. These guys have two. Sorry. And I'll note some other things, but basically these guys are cylindrical, these guys are flattened, these guys have two pairs of legs per segment on each body side. These guys have one. So yes, they do have more legs, and they're all uniramus. Okay, let's go to the next one. So the next are the crustacea. So these include things like crabs, crawfish, copepods, barnacles, lots of sea things that we love to eat. They also have two tagmata, the cephalothorax and the abdomen, right? Just like the chelicerates. Most of their appendages are uniramus, but they have some biramus appendages. Okay, and I'll show you biramus in just a minute. And they usually have two pairs of antennae. So here are some examples. We have crabs, we have barnacles, we have pill bugs, or at least that's what a lot of people will call these. Copepods, these are marine organisms. And of course it includes shrimp and lobsters and things we like to eat. So there's two pairs of antennae, and usually there's one short and one long. And you can kind of see that here. One long set, one pair of short ones. This is a classic arthropod that's usually brought up as an example of a crustacean. By the way, notice one set of antennae here, shorter ones here. So they have two pairs of maxillae plus a pair of mandibles. And I'll show you some images of this in lecture. There are some images in your book. Suffice it to say, these guys, again, lots of mouth parts. They have pairs of appendages on each body segment. So remember, cephalothorax, abdomen. And you can see all kinds of like appendages everywhere. And you can see the metamerism here, left over. So these guys have some biramus appendages, not all. So remember, biramus means they're branched. So it's primarily, on this guy, it's the swimmerets that you're seeing that are branched appendages. So this is a close-up diagram of the appendage. So this is where the branching comes from. So one part's called the exopod, the other part is called the endopod. These are the branched part that stick out, and you see a little bit of this. And the gills are actually on the inside of the body up in here. So they actually use these legs both to hang on to their young and to aerate the eggs, as well as to breathe with, strangely enough. And if you look on the underside, so these are the swimmerets here, and this is where they're branched. You can see where there's the two bits here on the end. Okay, and then to go into the last group, we have the hexapoda. So these are all the insects, okay? Insects and some other things too. They have three tagmata: head, thorax, and abdomen. Okay, so you can easily see that: head, thorax, abdomen. All of their appendages are uniramus, so they're unbranched, and they have one pair of antennae. So if we go into a little bit of detail with them, here's a typical sort of roach, bees. These are leafhoppers. I think they're really cute. Of course, we have a praying mantis that's eating some sort of a grasshopper-looking thing. This is a flea, dragonfly, dung beetles, also my favorites as well. So all exemplify the hexapoda. So they have one pair of antennae, but let me tell you, the variation in antennae in the hexapoda is unbelievable. And probably the hallmark of all of them are the differences between moths and butterflies. So these are butterfly antennae, so they're usually clubbed. So they have what looks like a really thin shaft and these little clubbed ends. And then these are sort of classic antennae for moths. So feathery, sort of branched looking, very good at chemosensation over long distances. And here you can see the antennae on bees, right here in the front. And then here on a ladybug and on ants. So antennae are incredibly useful for picking up vibrations, currents, but especially for chemosensation. They have three pairs of legs, 1, 2, 3, all uniramus. Three tagmata, so head, thorax, and abdomen. And they often have two pairs of wings, but not always. So clearly some don't. Here you have a classic dragonfly, two pairs of wings, fore and aft. Same thing with butterflies. Actually, that's a Luna moth. And then here you have beetles. So usually they have these coverings called elytra that cover up their wings because their wings are very delicate. And this is a real live beetle. It looks like it's painted gold, but it's real. And here's just another view with the elytra held up, and you can see the wings. So these guys, the hexapods, also have Malpighian tubules. So commit this to memory, and you'll see these in lab as well. So now don't get these mixed up. So here's the Malpighian tubules. Remember, they're outpocketings from the intestine, and they extract water from the digestive system to help maintain water balance. But you will see these structures when you do your grasshopper dissection as well. But these are gastric ceci. So these are little outpocketings of the digestive system that hold lots of bacteria, and they help to break down the food, to help digest food for the grasshoppers, crickets, and lubbers, and things like that. By the way, one thing you should really notice here is in the digestive system, and they're not showing it in full detail, but you can see how regionalized it is. So you have a mouth that goes into a pharynx, You then have a crop, which is is sort of akin to a stomach. You have the proventriculus and then these gastric ceci, so you're starting to have these specialized digestive organs and regionalization of the digestive system. Here's the midgut. Of course, you have the malpighian tubules. You have the hind ends, or the colon. And there are other organs that are associated with the digestive system in hexapods as well. Okay, and then, oh my God, talk about diversity of parts. So mouth parts galore. So you're going to see this in lab, and we're going to do more of this in class. I'll show you some videos. But look at all these different parts of the mouth, right? So here's some of the mandibles, here's the maxilla, and some of the labial palps. So within the hexapoda, you have all kinds of mouth parts. So you have chewing mouth parts, you have sucking mouth parts, and these are like sucking and piercing. So the mosquito's got to get into you and then suck out your blood. But there's different kinds. So butterfly also has sucking mouth parts, but this is more like a straw, and it's taking out. It's not piercing. These are piercing and sucking. Butterflies are just sucking. And then with horsefly, they actually have sponging mouth parts. So they have these labial lobes that are basically for like picking up either rotting juices or your sweat or all kinds of liquidy goodness that they can digest. So we talked a little bit about trophic breadth and development. So there's two different kinds I'd like you to be familiar with. What's called holometabolous is also known as complete development, where you go from an egg to a caterpillar, so you have one sort of life form here, and then you metamorphose through into your final life form. So basically, the adult form looks completely different from the juvenile form. Hemimetabolous is known as incomplete development. And so what that means is you hatch out as a miniature adult, okay, but you're only really lacking size and wings in this case, right? So you grow up, you get bigger, you start to look like an adult, and then you get some wings. So this is referred to as incomplete development because you're not going through true metamorphosis and true, change. Whereas here you're going through, huge change. And these are both really successful forms of development that are exhibited in the hexapoda and have allowed them to exploit different resources at different stages of their lives so that they're not competing with the different life stages. Okay, that's all I have for you now. Be sure and fill out that worksheet and have it ready for class on Wednesday, and I'll see you on Wednesday.